el formato virtual a través del cual podemos atender las conferencias magistrales y las presentaciones de los trabajos de los primeros a través de medios digitales, así como interactuar con ellos por medio de las herramientas provistas por el comité organizador. Este cambio de paradigma nos llevará a adquirir nueva experiencia en el empleo de las tecnologías de la información y comunicación en la organización y realización de este tipo de reuniones académicas y se convierten además en una alternativa muy valiosa para dar continuidad a la celebración de nuestro Congreso, muy a tono con la era digital en la que vivimos hoy en día y las tendencias actuales en la difusión del conocimiento. Pero además, en esta ocasión, como dije al principio, también como parte de los festejos académicos del 50º del ICAT, hemos conjuntado la celebración del Congreso SOMI 35 con el primer Simposio Nacional de Biosensores, en el que se explorarán nuevas áreas de oportunidad para fortalecer el diseño y desarrollo de estos dispositivos. Los sensores presentan un número muy amplio de aplicaciones en el monitoreo del ambiente, detección de enfermedades, en la industria alimentaria y farmacéutica, entre muchos otros. En México se cuenta con un importante número de grupos de investigación enfocados en el diseño de estos dispositivos, así como en su desarrollo, por lo que hemos decidido llevar a cabo esta primera reunión en conjunto con el SOMI. Así, este encuentro permitirá también analizar y discutir parte del avance que se presenta en el área de biosensores en un ámbito nacional e internacional para fortalecer lazos académicos en pro del conocimiento científico. De esta manera contaremos con 6 conferencias magistrales de expertos de primer nivel en instrumentación y biosensores, provenientes de instituciones académicas de México, Canadá, España y Suecia, las cuales serán dictadas a lo largo de los 3 días de celebración del Congreso y el simposio, así como con la presencia de alrededor de 90 trabajos de los participantes en formato de video, seleccionados por el Comité Científico Técnico entre 12 campos distintos y alrededor de 50 áreas temáticas de instrumentación y biosensores. En esta ocasión, los participantes provienen de casi 30 instituciones. A todos los participantes, nuestro reconocimiento y gratitud al hacer posible la realización de estas actividades a través de sus interesantes contribuciones. También quiero reconocer el trabajo realizado por todos los integrantes del comité organizador del Congreso SOMI 35, integrado por académicos de los diferentes departamentos del ICAT, y agradecer particularmente, por motivo del 50º del ICAT, a todos aquellos que desde hace 41 años han realizado y mantenido vigente esta actividad, en especial al maestro Gerardo Ruiz Botello, así como a los impulsores del primer Sinabios, sin olvidar el importante apoyo brindado y la contribución al Congreso que realizan cada año la Unidad de Cómputo, la Coordinación de Difusión y la Secretaría Administrativa del ICAT, así como a quienes apoyan en la transmisión de las actividades a través de los canales de YouTube, del ICAT y del SOMI y de otras redes sociales, y a quienes se hacen cargo del diseño de imagen del Congreso. Por supuesto, merece particular mención el trabajo de selección de los trabajos que cada año realiza el Comité Científico Técnico del Congreso a través de la participación de especialistas del ICAT y otras instituciones nacionales e internacionales para conformar un programa de calidad e interés para la comunidad instrumentista participante, así como el trabajo de edición de las memorias por parte del personal académico de diferentes departamentos del ICAT. Finalmente, quisiera expresar mi particular y profundo agradecimiento a la Coordinación de la Investigación Científica de la UNAM, sin cuyo apoyo a lo largo de los años y en particular en esta ocasión, estas actividades académicas no habrían sido posibles. Les deseo y les auguro el mayor de los éxitos en la celebración del SOMI 35 Congreso de Instrumentación y el primer Simposio Nacional de Biosensores, ambos en su primera edición virtual, con la expectativa de seguir contando con su interés y entusiasta participación en las actividades por venir. A todos, muchas felicidades y mucho éxito. Muchas gracias.
Muchas gracias, doctor Zanella, por este alentador mensaje. La verdad es que es una inyección de ánimo y de energía para llevar a cabo este congreso y para continuar con él. Sí, así como con el nuevo esfuerzo que empezaremos a hacer en el ICAT a partir de hoy a través del tema de los biosensores, con el simposio biosensores, ¿verdad? Muy bien, enseguida quisiera yo presentar a la doctora, de nuevo, a la doctora Claudia Rodríguez, en este caso ya en su calidad de moderadora de nuestra primera conferencia magistral que va a dictar el doctor Daniel Ailey, precisamente proveniente de Suecia. Entonces, sin más preámbulo y para darle paso a la presentación del doctor Ailey y después al desarrollo de su conferencia, le pediría yo a la doctora Claudia Rodríguez que por favor se haga cargo de la moderación de la siguiente conferencia. Muchas gracias.
Hola, muchas gracias. Gracias al doctor Rodolfo por igualmente, de acuerdo al doctor Gerardo, en concordancia con él, con este alentador mensaje, sobre todo porque es la primera vez que se hace esta reunión en el área de biosensores a nivel nacional. Agradezco mucho todo el apoyo y muchas gracias, Gerardo. Y bueno, buenos días a todos, buenas tardes y bienvenidos por todos los participantes y los que nos escuchan. Vamos a dar comienzo a las actividades de este día con la conferencia magistral del doctor Daniel Ailey. Le voy a dar en inglés su introducción. It is with great honor that I may now introduce to you Professor Daniel Ailey. He's an expert in the area about sensors with a master's degree in biological engineering, obtained a doctorate in sensor science, both at Linköping University, Sweden, carried out 2 postdoctoral studies at the Institute of Biomedical Engineering, Imperial College, London, and Nanyang Technological University, Singapore. He currently works at Linköping University, Sweden, as a professor in molecular physics. He is head of the Molecular Material Laboratory and the Division of Molecular Physics, Department of Physics, Chemistry and Biology, is the supervisor of graduate doctorates, has obtained grants and awards, such as Future Research Leader by SSF, Wallenberg Academy Fellow, KAW, the Vinova VR Joint Column Biologics, the Nobel Nordic Prize for Sulfate and Colloid Chemistry, the Amberska Awards, Royal Swedish Academy of Science, participate as a member of the editorial board of Scientific Report, member of the editorial advisory board of Peptide Science, member of the board of the Strategic Research Area of the Swedish Government in Material Science on Functional Materials at Linköping University. He is a grant evaluator at the German Research Foundation, Swedish Foundation for Strategic Research, expert reviewer, Swedish Foundation for Strategic Research, external grants evaluator, and reviewer in many journals with high impact as Nature Chemistry and other important journals. He is a member of the Swedish Chemical Society, the Scandinavian Biomaterial Society, the European Biomaterial Society, and the Material Research Society. Welcome, Professor Daniel Ali, and welcome to this meeting. And thank you for participate in this event.
Thank you very much for that very kind introduction, and yeah, it's an honor to be here, yeah, really looking forward to share some of our work with you today. So let's see if I can share my screen. Great.
Yes, it's okay.
Perfect. Just give me a second. See if I can. Just need to minimize the videos here. Okay, so thank you again for that very kind introduction. I will talk to you today about our work on nanoplasmonic biosensors for various clinical and industrial applications. So as you heard, I'm a professor at Linköping University at the Division of Biophysics and Bioengineering. So just to give you an idea where this is in the world, Linköping is Sweden, on the Scandinavian peninsula, about 200 kilometers south of Stockholm, the capital of Sweden. So we're still quite a bit south of the Arctic Circle. But winter is coming, apparently. So I wish I were in Mexico. I heard you had a slightly warmer weather than we have. So Linköping University, it's one of Sweden's larger universities. It has a bit more than 30,000 students and 4,000 employees. In the biosensor community, Linköping University is, I would guess, most well-renowned for these three gentlemen that have affiliations with our university here. Professor Tony Turner was in the same department here as I, and he's, as you know, one of the front figures of the biosensor community. And then as well as Professor Ingmar Lundström and Professor Bo Lidberg, who was pioneering the work on optical biosensors based on surface plasma resonance, which is very close to my field as well. So biosensor, as you're well aware of, is an analytical device for detection of chemical substances, which we typically refer to as analytes. The devices, they combine a biological component with a physical-chemical detector. That's what's unique to biosensors. And this biological component, it provides means for specific recognition of analytes. Whereas the transducer, this detector, then converts this biorecognition event into a signal that we can detect. The bioreceptors, or ligands, as we often call them, are, it can be essentially any kind of biomolecule, such as DNA, proteins, or peptides, or even entire cells or cell fragments, or even tissues. The transducer is usually converting this interaction to an optical or electrical or electrochemical or signal, or measures changes in heat due to these interactions. And then typically we also require some sort of amplification of this signal for us to record it properly. And in my work, I primarily, interested in optical sensors and using proteins and peptides as ligands or receptors. And my work is, as I mentioned, quite much inspired by the seminal work of Professor Lidberg and Lundström on surface plasma resonance. SPR, or SPR-based biosensor is probably the most widely distributed or used biosensor, at least for research purposes today, and they come from many different companies. But the work of Lidberg and Lundström was commercialized by the company Biacorno, which is now owned by Sutiva, formerly GE Healthcare. So this is a generic optical biosensor technology for biomolecular interaction analysis, which, let me see, which essentially, it's an optical technique which uses the properties of propagating surface plasma at the metal dielectric interface, such as this gold surface that we see here. And then we can turn this into a biosensor by immobilizing biomolecules on the metal, interfacing the medium around the metal, and then detect ligand-analyte interaction as it occurs on the surface. And essentially what we monitor are changes in reflectivity as we change the refractive index in the vicinity of this surface. So shift in the reflectivity of light irradiated at the surface. And we can do this detection in real time, so look at analyte ligand interaction as it happens on the surface. These propagating surface plasma, that's are essentially collective electron oscillation that are induced by this visible light, and which essentially result in an evanescent field that propagates into the surrounding medium. That's what's tried to be illustrated here. And that evanescent wave essentially propagates around or reaches out roughly 200 nanometers from the surface into the surrounding, which makes this as a fairly surface sensitive technique, so we can monitor what goes on close to the surface here. And from being a nice to have instrument in companies working with drug development, this has changed quite dramatically with the last 10 years or so to a must-have instrument in drug development. But these instruments, they are quite pricey. They are rather complicated to use and to interpret the data, so it's required quite a lot of expertise and also to maintain these instruments. And due to the fairly large sensing depth, they are also recording quite a lot of the refractive index changes occurring a bit further away from the surface than we are necessarily interested in, giving rise to a large bulk refractive index shift. And this, in combination with some other aspects, make these instruments very sensitive to temperature fluctuations. And they are essentially optimized for pure samples, low volumes, and relatively low flow rates. But excellent for research purposes, especially in drug delivery. But what my research is about is essentially to see if we can make cheaper, more robust, and flexible optical biosensors with similar performance as you can essentially obtain from surface plasma resonance based transducers. And this made us look into the field of localized surface plasma resonance. So essentially a very related phenomena to SPR, but using metal nanostructures instead. And when having metal particles that are small in size, and small in size typically means less than 100 nanometers, then we can much easier induce collective electron oscillation. Essentially, we don't need any complicated optical setup to do this. So we can just irradiate the particles and we induce these collective electron oscillations, which results then in a lot of absorption of the visible light as well as scattering. And we can see this as a quite pronounced extinction band in the visible wavelength range. For particles this size, this occurs in the green wavelength range, which makes the suspension of these particles look red. As you can see, there's literally no interaction with lights here at this wavelength. And the position and intensity of this LSPR band depends, just as in the case of SPR, on the dielectric properties of the vicinity of the particles. So if we, for example, immobilize or adsorb some sort of biomolecules on these particles, we'll obtain a change or a redshift, typically in the plasmon band. So essentially what we are detecting here is changes in refractive index, just similar to SPR. And then we also have this term here in this equation that determines the magnitude of this SPR shift or plasmon band shift. That's what we refer to as refractive index sensitivity. And we'll come back to that during this talk quite a lot, what we can do to essentially improve this, to increase sensitivity of sensors based on LSPR. We can also look what other aspects in the metal nanostructures that influences the optical properties here of this, or sensor performance of sensors based on LSPR. And one such factor is the size of the particles, and that influences the LSPR band quite a lot. And of course also the dielectric function of the metals used, so the materials essentially. I already mentioned the surrounding medium, and of course also the shape of the nanoparticles that we're using, or the nanostructures, and also how closely they are spaced if we get optical coupling between the particles. And optical coupling between particles is a phenomena that's been studied for quite some time, I would say. At least the earliest, to my knowledge, record in the scientific literature is a paper by Michael Faraday from 1857, where he describes this phenomena where you get the colorimetric shift of particles aggregating. And he also concludes in this paper that this is actually small pieces of metal that was so tiny that he couldn't see them by his powerful microscopes, but just by doing careful scientific work, which is really interesting to read about. Anyhow, also a few years later, in 1912, Carl Friedrich Lange published a paper on using this phenomena as well, the aggregation of particles and the optical coupling and the resultant color change for diagnostics. So also, to my knowledge, the first nanoparticle-based biosensor. And he used this phenomena for diagnostics of patients with syphilis. So essentially taking cerebrospinal fluid from these patients and subjecting them to gold nanoparticles. And if you had syphilis, they started to aggregate. If you were healthy, they did not, for some reason. And this test was used for quite many years in the clinics. But it can also be used for more specific biosensing, as we think of biosensing, where you actually have a biorecognition element that triggers aggregation or de-aggregation of particles as a result of binding of the analyte. And this can be done in many different ways. And the only limitation here is essentially, or there are a few actually, but one of the main limitations, that you have to end up with particles that are close enough to get this optical coupling, which means that they usually need to end up less than a separation less than this diameter of the particles. We have, and many others have investigated various ways to do this, use this phenomena for biosensing. And this is one example from our work, where we have been developing sensors for lipases, which is a large group of enzymes that hydrolyzes lipids, and also very highly interesting and relevant biomarkers for a range of diseases and also drug targets. And lipases, they hydrolyze lipids, so the substrate is, of course, a lipid bilayer then. so we essentially, to go about this, encapsulated a designed peptide inside liposomes, and this peptide is designed in such a way that it can trigger aggregation of gold nanoparticles that are functionalized with a complementary peptide, and these particles are kept outside the liposomes. And then when we add phospholipases, they start to degrade the lipid bilayer. Eventually the polypeptides will be released due to this degradation process, and this triggers an aggregation of these particles. And we can monitor this as essentially a redshift of the plasmon band, which is shown here on this axis, and then we monitor this process over time, and we can see that we can get slightly different magnitude in shift depending on concentration, but above all, the higher the concentration or activity of this enzyme, the faster is this release. And this were then later also developed further together with collaborators into a device for diagnostics of pancreatitis, where you have quite a large overexpression of lipases. And this is an acute pathological condition which requires immediate attention by a physiologist, or you need care within hours, essentially. So a rapid diagnostic here is key for the correct treatment. And with this lateral flow device based on this concept, it was possible to diagnose pancreatitis in patients in serum samples within 10 minutes. So colorimetric sensors has the advantage that they are simple, and the readout is also very rapid and simple, because you don't need any elaborate equipment. Typically they are naked eye detection. You can use them to monitor enzyme activity, as you saw in these examples, but they require elaborate assay design, and there's no generic ways to make these assays. You have to redesign them for each analyte, which is quite tedious. And you also have some issues with nanoparticle stability, since colloids are not thermodynamically stable. They tend to aggregate over time, or due to unspecific adsorption of proteins. So that's why we also have paid quite a lot of attention to refractive index sensing. So essentially approaching the same concept as used with SPR, just monitoring small changes in refractive index when biomolecules binds to the surface of nanoparticles. Because this is more generic sensing strategy, you don't have to consider nanoparticle stability, and you can have the particles immobilized on the substrate. The downside is that the shifts here, the optical shifts, are very small, and you typically require a spectrophotometer of some sort. If we compare the bulk refractive index sensitivity of LSPR and SPR, there is an order of magnitude difference, or orders of magnitude difference, depending on what particles we use here. And one of the main reasons for this, as I mentioned, in SPR we have a quite large sensing depth, so we can detect interactions occurring all the way from the surface to 200 nanometers roughly from the surface or the interface, whereas with particles the sensing depth is much more restricted, typically around 20 nanometers, as we can see when we just, if you adsorb polymer layers on top of the particles here, we can see that the shift decreases quite dramatically. So this is both an advantage and a limitation, as I can show you. The advantage, I would say, is that if you look here, most of the interaction actually occurs within this, on this distance from the surface. So whatever goes on here, we're not typically really interested in, and that can actually just contribute in making the signal from the biorecognition event more difficult to interpret. So if we instead just look at what happens in the vicinity of the surface within 50 nanometers or so, we can see that, yeah, then there's not such a huge gap between these simple spherical 50 nanometer particles and SPR as the method. And we can actually, by using some more sophisticated nanoparticles, such as these gold nanoframe protected silver nanoprism, actually outcompete SPR, at least within the first 20 nanometers or so from the surface. So obviously, we have quite a large dependence here, as you can see, on the refractive index sensitivity on what kind of particles we use. And we can compete with SPR here in this instance here. So these gold frame silver nanoprints, they are relatively good with respect to sensitivity. But we see that there is also other particles that can perform rather well. Some are less complicated to synthesize as compared to these prisms. And the reason for having this gold frame, I should say, is that silver in general is, although it has good sensing performance, it's very sensitive to oxidation. So in order to protect retain the morphology or geometry of these prisms, we protected them with this gold layer. But the main reason why we see differences here in morphology or differences in sensitivity for nanoparticles with different shapes is because they have different position of the LSPR band. And if you plot the position of the LSPR band, the maximum, as we have done here, against the sensitivity, you see that we can essentially have a straight line here. So there is a direct correlation between the position of the LSPR band and the sensitivity, and the higher the refractive index sensitivity, the higher the sensitivity. So, and then you might think that, okay, so if we could, we can obtain this by elaborate synthesis of particles, but we could also have a shift in the plasmon band by aggregating the particles, and that's true indeed. We can redshift the plasmon band by just unspecific aggregation of the particles, but as you can see, we also get a broadening then of the plasmon band, and that makes it more difficult to follow changes in the plasmon band. So, and in order to essentially take that into account when we judge the sensing performance of an LSPR device, we also need to look at the width of the plasmon band. And we defined this so-called figure of merit as the sensitivity divided by the full width half maximum. So the more narrow the plasmon band, the better typically the performance of the sensor. But we can still employ plasmonic coupling to redshift the plasmon band if we do this in a more defined way than just unspecific aggregation. And we can do it by essentially making satellite structures of smaller and larger particles separated by a well-defined distance using polymer layers, as in this case. And we can see that going from one very distinct plasmon band to essentially split this up into several different plasmon modes due to this plasmonic coupling. And we can see that we redshift the second plasmon mode here more the closer the particles are to each other. So just one polymer layer, we have a very well-defined second plasmon band here that we can use for sensing. And this actually gives us an increase in sensitivity with a factor of 2 to 3 compared to the individual particles. And again, primarily an effect of redshift of the plasmon band. And in this case, you probably noticed that we also use the immobilized particles, and that's one of the key advantages with a refractive index sensing here, that you don't have to work with particles that are suspended. And there are various ways to immobilize particles, and you can use all sorts of different substrates, such as glass, silicon, metal, polymers. And then you can tune the surface chemistry of these substrates, for example, by plasma treatment, or by self-assembly of monolayers with different terminal functionalities, or just use polymer adsorption in order to bind nanoparticles to the substrate. And then, of course, you can also adapt your nanoparticle surface chemistry to work well for this purpose, as well as modulate the ionic strength to, for example, increase or decrease packing of particles on the substrate. And these are some examples of sensor surfaces that we've been working with, primarily quite a lot with gold particles on glass. And so this is what it can look like in electron microscope, or gold nanorods on plasma treated silicon oxide, or gold nanoparticles adsorbed to a nanocellulose substrate, or gold particles adsorbed or immobilized on a plastic substrate. So there are many options here. So depending on your applications or what you want to study, you can essentially pick and choose whatever substrate that works for you. But there are some things you need to take into account, and also when choosing substrate particle combination. And as we can see in this data here, here we have spherical silver nanoparticles, and if we immobilize them on a glass substrate, there is essentially not much of a difference in sensitivity, where if we go for cubes or plates, also silver-based, we see a quite dramatic decrease in sensitivity. And these particles were originally more sensitive than the spheres here, but due to this effect, this difference is actually decreasing quite a lot. And this is due to essentially the high refractive index of the substrate, resulting in some of the electric fields are distributed to the materials instead of probing the surrounding medium. And the larger the contact area between the particle and the substrate, the larger is this substrate effect. So we should try to keep this at a minimum then, or use, if this is critical, low refractive index substrates. And these differences are typically what we are talking about now with these particles. We typically rely on a spectrophotometer to monitor these changes in refractive index, but we can also, with some tricks, make them visible by naked eye. And that requires that we take the physiology of human vision into account. So I mean, we can boost sensitivity by redshifting the plasmon band to have particles that give rise to or produce a large shift in the plasmon band as a result of fairly small refractive index changes. And this is another example of highly performing particles for this particular purpose, gold star nanoparticles, where we get a 40 nanometer shift from just roughly a 30% of monolayer proteins adsorbed on the particles. But still, we can't see any changes here by eye before and after absorption of these proteins on the surface. Whereas if we look on these particles instead, this is a gold-silver alloy particle, spherical. For a much smaller refractive index or LSPR shift, we actually can see this difference by the naked eye. And this is due to that this transition or this shift happens just as a transition between two colors, where our eyes are essentially very sensitive to these changes. So here we can actually, if we take this into account, we may not even need a any kind of spectrophotometer. We can use cell phones, for example, to look at these color differences. But in most cases, we have access to a simple spectrophotometer of some kind. And for example, in most labs, there are plate readers available. And plate readers, they use well plates as a carrier of samples, which comes in various shapes and forms, but they are always made of plastics, typically. And since we can immobilize particles on plastics, we can turn these well plates into LSPR sensor surfaces. And that's what you see here. The pink color here comes from particles immobilized on the plastic of these well plates. And in this case, we were interested in making a sensor for detection of proteolytic activity. Proteases are highly important in numerous physiological processes, but are also very interesting biomarkers for example, cancer and other inflammatory diseases, and also in infections. So as a model protease, we use trypsin, which essentially hydrolyzes or digests any protein. And as a protein or as a substrate for trypsin, we use the casein, just physisorbed on these particles. So a very simple setup, and then exposing these sensors to trypsin, we can see that we get in this case, a blue shift of the plasmon band. So this shows the LSPR band during the casein immobilization or functionalization of the particles of casein. We have a red shift, and when we degrade the casein, we remove, of course, mass from the surface, which we see as a slight change in the LSPR band and intensity. But sufficiently large to see this quite clearly with a plate reader. And using this simple strategy, we can detect fairly low concentrations of trypsin. But it was not trypsin that was our main target in this case, and we were interested in monitoring proteases involved in a proteolytic disease in the oral cavity. So this is probably something many of you have experienced, the bleeding gum. And this is usually what you see when you're suffering from periodontal disease, which is a bacterial infection, a chronic inflammation of the gum, which eventually leads to tooth loss. And it's also associated with high risk for Alzheimer's disease and cardiovascular disease. And the main pathogenic driver in this disease are the bacteria Porphyromonas gingivalis, which secrete gingipains, which is a group of enzymes that degrades the tissues. So this disease is typically diagnosed, as you see here, by probing the dental pockets, the gingival pockets, and that's essentially when you can. I mean, that's the late stage of the disease, essentially, when it's harder to do something about it. So the dentists are looking for simple and rapid ways to detect this biomarkers at an earlier stage. And we tested the same approach here for gingipains, and we're able also with the plate reader to detect clinically relevant concentrations of gingipains, both KGPs and RGPs, in essentially concentrations similar to what can be used or is required for early diagnostics. But in addition to diagnostics, we are also interested in exploring LSPR biosensors for bioprocess monitoring, and in particular processes aiming at production of biopharmaceuticals such as therapeutic antibodies, which are quite new kind of drugs that are essentially changing the entire, currently the entire pharma industry now, as we get better tools for developing new biopharmaceuticals. But the problem here is that these processes, I mean, here we rely on culturing of cells and cells to express these complex molecules, and then they need to be purified through several steps. And these processes are associated with very high costs, and they're quite complex, and it takes very much time, essentially, to produce these drugs. And one of the main reasons is that there is a huge, there's a complete lack of methods to monitor both the actual product being produced and also some key contaminants during this process, making it very difficult to intensify and make these processes more continuous and to make them more automatic. So our ambition here is to essentially facilitate work to intensify bioproduction by essentially enabling inline quality control and detection of, for example, product titer, product variants, or contaminants. And we do this by essentially using sensors similar to what you see in this illustration here. So flow cell, very simple, combined with an optical fiber that's connecting to light source and a detector. And then we have a removable sensor chip that is functionalized with particles. And it looks like this. So stainless steel flow cell, inlet, outlet, and optical fiber. And in here you have the sensor chip. And with this fairly simple device, we can outcompete essentially current techniques for at-line detection of, for example, titer, both with respect to sensitivity and dynamic range, but above all, the speed. I mean, here we can detect or get a result within minutes on the product titer throughout the entire bioprocess step, from the complex soup of biomolecules with very low titer concentrations in the upstream process throughout the purification and clarification steps. And very good correlation to these other methods that typically take hours to get the same results. And also we can, at least when using it at line, get information on binding kinetics, so similar to what you can do with the Biacore instrument. And we can also quite easily adopt the instrument to monitor analytes over a quite wide dynamic range, from essentially nanomolar up to millimolar. And we are also exploring inline applications, for example, to monitor product breakthrough in chromatography steps, where these sensors, as you can see in this photograph here, is connected to a chromatography equipment on pilot scale. So roughly flow rates around 150 milliliters per minute. And to detect essentially when the product saturates the column, and then you can use this essentially to optimize the usage of this extremely expensive chromatography medium. This is currently not possible to do with UV chromatography, since there are so many other proteins there. The signal in the UV spectrophotometer is saturated, whereas here we see a clear binding when the product essentially is or when your column is saturated and the product is starting to go through. And we can see this even better if we zoom in. There is no essentially signal at all in the UV, whereas with the LSPR we can clearly distinguish when the product is going through the system. But there is still definitely a lot of room for improvements here. This is a photograph of a glass substrate that has been functionalized with gold nanoparticles. And you can see this faint pink color comes from the particles. And here we actually have fairly high density of particles there. You can, of course, squeeze in more particles there. It's definitely possible. But then this starts to happen, that we get a broadening of the plasmon band and the redshift. And that kind of have a negative impact on our figure of merit and the sensing performance. So we can't really improve the signal intensity by just squeezing in more particles here. So we started to look on possibilities to instead increasing number of particles in the third dimension by using three-dimensional substrates. And one such substrate that caught our attention was nanocellulose hydrogels. So these are essentially biopolymeric materials, water rich materials, but can be produced at with really high quality and reproducibility. And what you see here in the bottom of this cuvette is one of these membranes here, cellulose membranes. And we wanted to develop an approach where we use self-assembly of particles in here, so we can start with well-defined particles. There's been a number of papers published on attempts to synthesize particles in nanocellulose, but then you don't have the same control over particle size and composition and shape as you can do if you prepare particles separately and then adsorb them in the cellulose. So after some optimization, we were able to essentially make all these particles go from being suspended to being adsorbed in this membrane. And as you can see here, we end up with a substrate that is not faint pink, but bright red. We can also increase this intensity quite much more, but then you start to get a broadening of the plasmon band. But in this case, we have a very well-defined plasmon band. And this is due to the particles being fairly well distributed within this three-dimensional matrix, both on the surface but also throughout the entire volume of this hydrogels. And when we compare the intensity between these three-dimensional materials and the two-dimensional, we can see that there's a quite substantial difference here. But if we normalize the data here, we see that there's no shifts or anything or broadening of the curves. So there are still essentially we have the same figure of merit, and this is also due to there's no differences in refractive index sensitivity here. So what this means is that we increase the signal, or we increase the signal to noise. And for the 2D surfaces, we have a signal to noise ratio roughly around 250, whereas in this case, it's substantially higher, so around 450. And what this in practice means that we have a much better reproducibility when we measure things, because there is less influence from noise. And if we just look at an example here where we adsorb bovine serum albumin on the surfaces here, we see that there is quite small variation here in the pink three-dimensional data here, whereas in the blue here, two-dimensional data, there is quite much larger variations due to the higher signal to noise. But this technique with the self-assembly here of the particles in the membranes, we can also essentially immobilize all sorts of different kinds of particles, or a combination of particles as well, to do more elaborate nanoplasmonic materials. So that we thought was quite interesting, but something that really caught our attention was what happened when we subjected these composite materials to a bit of mechanical pressure. Then we saw that we got a change in color similar to what we see when we aggregated particles. But when we looked at these in the electron microscope, the particles were still fairly well separated. There was no difference here. So this is essentially just an effect of pressing or compressing the material and making the particles immobilized on individual fibrils to come close enough for us to get plasmonic coupling. And we started exploring what could essentially influence this mechanoplasmonic coupling. So when comparing an uncompressed material with a compressed material, we see a quite distinct color differences. But if we immobilize a protein, or just by physisorption on the particles before we compress, we don't see much of a color change. So essentially, the proteins here can prevent the particles from being mechanically forced into contact. So by essentially using mechanical pressure here, we can sense what goes on the nanoparticle surface. And also reasonably then, the more proteins you have on the surface, the more efficient are these proteins then inhibiting this mechanoplasmonic response here. So we can also get a concentration-dependent response. And with that, I would like to thank all the people in the group that has done all this work, especially Olof Eskilsson, Robert Selegård, Toy Tran, and Anna Svärd, and also our collaborators and colleagues around the world, and funding agencies. And thank you for your attention.
Thank you very much, Daniel, for this very nice presentation. I think it's time for questions. We have some questions from the audience. ¿Hay alguna pregunta de la audiencia? No sé si en el respaldo nos puedan facilitar las preguntas.
Okay, I have a question.
Yes.
Thank you for your presentation. Very interesting, Daniel. Have you measured the stability of the protein mobilized? This is because, it's about all proteins, but especially for antibody in solution, these molecules are not very stable. But have you measured these properties?
Yeah, that's a good question. I mean, it's very important if you, I mean, you mean the proteins on the sensor surface, or, yeah, so I mean, like in the case here with the sensors we developed for bioprocess monitoring, we use protein A for detection of antibodies. To a large extent, at least, we use protein A, and that's a fairly stable protein. We can use these chips for, we typically use them within 3 days, but if we store them in fridge, it's reproducible over 3 days. But of course, this is something we need to look into if we can optimize storage conditions, and I assume that's possible. I mean, you can buy sensor chips from Biacore, for example, with protein A that last for years. So there are techniques to essentially preserve the proteins on the surface. But then we are also interested in using these kind of sensors to look at stability of other proteins. For example, antibodies, as you measure, they aggregate in solution, for example, or they can be degraded. And that's also something which is very interesting in the production of therapeutic antibodies, to follow this and get a measure on how much of the antibodies are aggregated or degraded. We are currently looking at differences in binding affinities, for example, depending on if you have an aggregate or a fragmented antibody, and we see some quite interesting differences in the binding to our sensor surfaces, just using protein A as a ligand. But then also, when it comes to stability of ligands, we are also developing our own ligands based on peptides instead of proteins, since proteins are inherently stable. They won't last forever. But peptides, I mean, we can make them synthetically, and they're much cheaper from that perspective. And they last for as long as you like. I mean, they don't typically have any secondary structure to quaternary structure that can be disrupted. But then, yeah, then you have to do some more kind of screening of affinities and do some sequence optimization. But there are quite many methods for doing that. In my lab, we work with a technique called one bead, one compound peptide libraries for development of new peptide binders, which is a fairly rapid screening methods for identifying new ligands.
No, it's okay. And where more or less where among of antibodies are in the same orientation when it's immobilized? All is 100. All the molecules are in the same orientation. I suppose that no, or more or less. No,
I mean, typically, depending on what method you use for immobilization, it's random. Typically, I mean, if we just rely on physisorption, which we can do, then very random. Quite often we have some self-assembled monolayer on the particles and use a conjugation strategy that typically targets like primer amines on the ligands. Then still random, I would say. When it comes to peptides, I mean, then we can introduce functional groups in the peptides, which we can use for directed immobilization. So that's another advantage. And we use, for example, click chemistry with A-side functionalized peptides and strained alkynes on the surface. Or we also use cysteine functionalized peptides quite a lot.
Okay, it's very direct, no, the immobilization, because it says the protein, for example, in enzymes are very important, no, to how is the region of the proteins that immobilize for the function, and you are direct.
Yeah, it definitely influences the performance. I mean, those ligands that are immobilized in a geometry where they can't bind to the analyte, they are just not contributing to the sensor response, essentially just occupying the area and reducing sensitivity of the sensor. So it's an important aspect. I mean, by protein engineering, of course, you can introduce various also tags to facilitate directed immobilization. But that's nothing we are working with.
Okay. And about the size of proteins, it depends of the proteins that you want to immobilize in these nanoparticles. It depends of the size of the nanoparticles, or is a standard size of the nanoparticles, and you can immobilize any size of proteins, or is there a limit?
That's also a good question. I mean, we primarily use just simple spherical 50 nanometer gold particles for most of our sensors, refractive index sensors, even though they are quite poor with respect to refractive index sensitivity, but they have a fairly good figure of merit since we can make them with a very well-defined size and size distribution. But then of course also the sensing depth here is, I guess, what is the main limitation. It's not like we don't have room for the ligands on the particles, but since the sensing depth is roughly 20 nanometers, if we would have a huge ligand, like an antibody oriented, like with a constant part directed towards the surface, it's kind of 50 nanometers in size. So then you're essentially just using the outmost nanometers in the sensing depth here for detection. So that's not ideal, actually. So the smaller the ligand, the better, actually. Then you get the more sensitive sensor.
Okay. Thank you so much.
Thank you.
Silvestre.
Interesting question.
Gracias, doctora Claudia. ¿Hay alguien en la audiencia o los invitamos a hacer alguna pregunta? ¿Alguien que quiera preguntar o compartirnos?
¿Me escuchan, doctor Mendoza?
Sí, doctor.
Ah, es que mandé una por el chat, pero no sé si la pueden ver.
No tengo
no se ve.
no se ve.
¿No se ve? Ajá, ok.
Y-
Ok, es en relación a las dimensiones de los sensores,
ujú.
¿sí? Si, típicamente, ¿cuáles son las dimensiones de los sensores que están en desarrollo? Y si esas dimensiones en un momento dado influyen en la relación señal-ruido. ¿Sí?
Daniel, Professor Gerardo Antonio is asking, what are the dimensions of the sensors that you are developing? And if the dimension can influence in the signal to noise ratio?
Yeah, that's also an interesting question. Currently, if we talk about the sensor chips that we use for bioprocess monitoring, they are a few millimeters in diameter. We actually started out this work with making fiber optical sensors for monitoring biomarkers in a mouse brain. So we first we tried to make them very small. We can make them, or very small. I mean, they're less than a millimeter in diameter, I would say, in that case. And that works, but it was very tedious to work with. It can influence sensitivity in at least when using these kind of fiber optical detector that we use, because we need to collect the scattered photon or the reflected photons. And with this setup that we use, that's a limitation. So we can't really go much smaller. But in essence, there is not so much you're not limited in size. Essentially, you can do refractometric sensing on a single nanoparticle using dark field microscopy. So we can go tiny if needed.
Okay, muchas gracias, doctor Gerardo. No sé si haya alguien más de la audiencia que quiera preguntar. Okay, I have not received another question yet, so we have 3 minutes left, but I would like to ask you if, have you tried to immobilize another nanoparticle shapes like a nanorods, nanocubes for example, in the polyester wells for this paper of Anna?
Let's see. Yeah, we do. And you can do this essentially using a similar approach. So it's possible. Using by plasma treating the plastic, you can make it slightly negatively charged to immobilize rods that are C-tab capped, as long as you wash away excess surfactants first.
Yeah, well, in that case, I think you need to do you need to exchange this detergent with a citrate. So I think we discussed once.
No, we can use C-tab. You don't have to exchange it. Actually, the C-tab, since it's positively charged, it facilitates the immobilization. At least from my experience, exchanging the C-tab is quite tricky. Perhaps you know how to do this.
Yeah,
I just end up with aggregated particles when trying.
Yeah, exactly. So, and in this experiment of in the well plate, so have you seen a change in the enzyme activity? Because you have the substrate on the particles and then you add the enzyme. So did you see some change in the enzyme activity compared when you do the proteolysis in normal conditions without particles?
When comparing to other methods for detection of the proteolytic activity, which we did use in this paper, I can't say we saw any differences, but we didn't look at that in detail. Actually, it was more focused towards actually just seeing how sensitive or low in concentration we could go. But it's an interesting question, actually.
Because I imagine that you can do the other way around, immobilize enzyme and then detect some ligand or some. substrate, isn't it? And see what happens as well.
True. Yeah, if it's for essentially drug development sensor purposes, you can do it like this. If you develop something that is supposed to inhibit, for example, enzymatic activity.
Yeah. Okay. Well, I think it's time for conclude this very nice talk. And thank you very much, Daniel, for accept the invitation. And well, I don't know if the Dr. Claudia have some other question, some words.
Only thank you very much so much for your interesting talk again. And we hope that very soon we can meet again. In the next few days, you will receive an acknowledgement for your participation. And thank you so much. And bueno, para todos los que nos están siguiendo, continuamos con las otras actividades que son presentaciones orales, y en la tarde, a las 4 de la tarde, tenemos la conferencia del doctor Augusto García. Por favor, bueno, estén atentos a los horarios. Los invitamos a seguir participando en ambos eventos, hoy, mañana y el viernes, ¿no? En la tarde vamos a seguir con otras conferencias magistrales. En la página del ICAT pueden encontrar ahí el programa de SOMI y de biosensores de ambos eventos, ¿no? Que es de manera conjunta. Muchísimas gracias. No sé si alguien más tiene algún comentario.
Muchas gracias, Claudia. A todos los participantes, Dr. Zanella, desde luego, thank you very much, Dr. Aili, for your nice talk. Al doctor Silvestre Mendoza por su intervención, desde luego, muchísimas gracias también. Doctor Zanella, muy amable, muchas gracias por el apoyo y por estar atento al desarrollo y al curso del Congreso. Y ya lo dijo la doctora Claudia, les invitamos a seguir en este momento el programa del Congreso ya con los trabajos que presentan sus participantes a modo de video. Recuerden que las preguntas las pueden enviar por el chat y les serán remitidas a los autores a sus correos electrónicos. Sí, les agradecemos mucho y continuamos con el programa durante todo el día. Tenemos salida a comer a las 2 de la tarde y recuerden, ya lo dijo la doctora Claudia, a las 4 una interesantísima conferencia también con el doctor Augusto García Valenzuela. Hasta entonces, muchas gracias. Continuamos.
Gracias.
Luego.
Thank you,
Goodbye,
Daniel.
Daniel.
Thank you very much.